The last part we have here is looking at basically putting it all together. That concept of summation, that concept of plasticity, we'll mention those here. When it comes to pathways and strengthening connections, it's all about what happens at the synapse. If magic happens and it excites, it excites. Okay. Our EPSPs. It could be magic that inhibits. Just as awesome. Okay. Just as awesome. But again, everything is what happens at that synapse. Integration, when we talked about it when you talked about it in anatomy, we had divergence, convergence, parallel after discharged, reverberating serial pathways. We had a number of these different ways that we talk about it from an anatomical standpoint. Many nerves converging onto one, or one nerve converging onto many different ones, or in a series when it comes to basically how you make memories. Reverberation, all of those things that we looked at. All of it has to do with what happens at the synapse, okay? How can we get this nerve to fire? By a lot of stuff converging, all right? And, hundreds, there can be thousands that can synapse on it, okay? Again, this is gonna allow us to be a hell of a lot more effective at getting something to contract and something to relax. So we'll see a lot of convergence when we talk about, motor control. Sensory signals work a lot on divergence, okay? Where an axon will be stimulated and it will go and spread out to a number of other nervous tissue, okay? And we see a lot of that with sensory signals in the brain with some of our special senses. General senses too, I guess. Okay. I guess if I told you to, get a thumbtack, put it right side up and smack your hand down on it, you'd feel multiple things. Pain, regret. Why did I listen to him? And those are all various different regions of the brain interpreting that information. Okay. So, depending upon, is it a convergent path or convergent type of pathway or divergent kind of pathway, it really just depends on the type of modality we're talking about, sensory or some motor activity. And that's all I'm trying to say here. And the same concept we'll talk about when we talk about plasticity here in a second. It's basically like forming memories in serial processing works like that. A to B to C to D. I strengthen all those connections and I make a memory a hell of a lot more effective. Okay. And that's kind of serial pathway. So we're going to see a lot of these as we continue on here throughout at various different points in this section. And in the next section, when we start talking about, skeletal muscle control and whatnot. I said that some of these synapses can be summative in nature. Those EPSPs, those IPSPs, okay, those graded potentials are summatory in nature. All right? We have two types of summation, and summation just means to add. Okay, there's an impulse. There's your EPSP. Well, that didn't get me to threshold. Didn't travel very far, dissipated, it didn't go anywhere. All right. But if I have one EPSP and then I have another nerve come in there, and before this thing can completely dissipate, I have another one come in there and it just bangs off from there. It's like, wow, boom, we can make it all the way across, get to the axon hillock, and ta-da, magic down the action potential. We have two types of summation. Those within space, okay, and those in time. Spatial and temporal summation is summation in space and summation in time. I'll put it here, in space, and this is in time. Okay. So let me go to this next picture here, okay? So here is a beautiful nerve. And you can see it has a number of different neurons synapsing with it. And all of these are releasing a neurotransmitter. And these are all summing up. These are all of these neurons within the space that are forming a synapse. All of these nerves within the space, I'm adding it up. Okay, spatial summation. This nerve is excitatory, this one's inhibitory, excitatory, inhibitory, excitatory. We add them all up, and was it enough to get to the axon hillock, to get to that trigger zone? I don't know. But that is summation within space. Summation in time, okay. Summation in time is involving only one neuron. Okay, information flowing down here, electricity coming down here, and then another one right behind it. Okay, and then another one right behind it. All right. So as this electricity has traveled down here and stimulated, there's another one that's coming down right behind it, a few milliseconds after it, and it adds up. Okay, excuse me. So this is successive waves down the same neuron. Okay. So temporal in time, T of temporal, T of time. Same wave coming down the same nerve, triggering the same spot. Spatial summation is the sum of all of the various different nerves within the space around the effector. And it's additive in nature. They can be excitatory, they can be inhibitory. Okay. If we have 5 excitatory and 7 inhibitory, you're generally going to say, yeah, it's not going to fire. We add it all up. Right? So when it comes to these nerves and when they converge or diverge, okay, are we converging a lot of them within the space, all synapsing on a individual nerve? That would be spatial summation. We have one nerve coming down and synapsing on it, and it comes down again and again and again. That's temporal summation. Right? Either way, the great thing about them, these summations, if they are excitatory, let's just say, it can be an additive effect. And again, I'm keeping the sodium in. As that sodium stays in, I can create that electrical flip-flop of charge, right? And that creates that electricity. It's allowed to travel across. Right? So the ability of these things to be additive in nature is called summation. Spatial or temporal, whatever the case may be. Plasticity helps us to create memories. It helps the nerve to fire easier. If I'm helping the nerve to fire easier, what is the only way we can help it to fire quicker, easier? Do we make this thing start here, here, or here? If I want to make this fire easier, do I want to get it to A, or do I want to get my baseline level to B?
B.
B. Okay. So now it doesn't take as much to get the threshold to fire that AP eventually. Okay. So when it comes to plasticity, it makes it more excitable. And by more excitable, I mean getting it closer to 0. Okay. It never goes above 0. Okay. It gets it closer to 0. All right. This is what is called long-term potentiation. That's a fancy way to say make a long-term memory. Long-term memory. Okay. This is the way we make memories. This is learning. This is making memories. Okay. And you see it in the hippocampus. The hippocampus is just the primary region where memories are formed and retained. Okay. But it, again, I mean, this is going to work in all regions where, repetitive learning, motions, actions are stored. Okay. So we'll see this in the cerebellum as well, because there are things involving motor control there, right? More involuntary motor, well, not involuntary motor control, but it doesn't require input from the brain. Okay. If I said, close your eyes, pick up your pen and write your name, hell, you could all do it, even though you weren't watching it. Why? Magic? No, because you've learned that pattern of how to move your hand and write your name in cursive. And that memory is stored in those learned patterns, which is in the cerebellum. What does that look like? Here is these potentiators here. Okay. Here, I have an action potential going down, and it can stimulate the release of even more. This would be presynaptic facilitation. This is modulating this thing and making it a heck of a lot easier for that neuron A to affect neuron C. When it comes to memories, this is hugely important. Okay. Makes this want to get out into this, to bind to these, to fire. So these neuromodulators here, and again, acetylcholine was one of them, but there are many of them. These neuromodulators, these various different ligand modulators, neuromodulators, help to make it more easier to fire. When I make it more easier to fire, again, opens more calcium voltage gated channels. More calcium in means more vesicles can fuse with the plasma membrane, more neurotransmitters in the synapse, more binding to receptors, more ionic changes occurring, and the easier for neuron C to fire. And this is how we make a memory. Okay. There's a lot of great books out there when it comes to helping you memorize stuff or remember stuff and call stuff. I'll see if I can pull this up. I'm going to see here. This is, I'm going to turn off my thing for a second here. I apologize. Now you can see my dojo. So here's this book. It's Understanding How We Learn, and it's built off a couple other books that I've read that basically looks at this model right here on the screen. Okay, there we go. It's a pretty thin book. It's a very easy book. I even got my kid to read it. He never reads anything. It's helped him a little bit, but it's a great job. It does a great job of kind of looking at some science that looks at how we potentiate, how we can make these connections within the brain a lot more effective and a lot stronger, so you can recall it. And it's all about looking at stuff, putting it away and forgetting it, and going back and looking at it again. And that struggle, right? And when I say struggle, put in quotation marks, but that effort, that work it takes for your brain to try to recall that memory. And it's like, oh, okay, so this is something that I'm probably going to have to remember. So it'll go in and, you'll make these connections, and it will potentiate and make it a little bit easier for it. How, then you may ask yourself, how in the hell does it stay like that? If I have to forget it a little bit, put it away and look at something else and come back to it, if I didn't stimulate it, wouldn't it just atrophy and go away? And that is a fascinating concept. And the way to bolster this activity, sleep. If you get 7 to 8 hours a night of sleep, there are opioids and there are a number of other various different chemicals that your neurons release when you're asleep to tidy up and clean up all this stuff. cleans up these connections because there's a lot of wear and tear going on the brain all day long, right? And they're finding that during your sleep, and only during your sleep, for roughly 7 to 8 hours, I think it was 7 to 8 hours. It was not less. I don't believe it was less than 6. So I want to say around 7, if I remember right. Those that slept for 7 to 8 hours, they found that those that got more of that length of sleep had more of a greater ability to remember and recall information. And so then they started digging in and trying to look at the chemicals that are being released at that time and seeing the activity in the brain. And there is a buttload of activity going on in the brain, but it's all there for maintenance, for cleaning up. And there are a number of chemicals that get released when you sleep here to help strengthen up these connections that you've been looking at and studying all day, to help facilitate those memories, to strengthen those memories, okay? And if you look at information and then, you put that away and then you start looking at, another piece of information, let's say, you're looking at your math class, okay? And you come back in a couple of days, you start looking at it, and you're like, okay, you're asking yourself questions like, dang it, I remember that. I remember that. Okay, I'm missing this one thing. And you go back and you reread your notes. You're like, okay, that to that to that. Okay, that's what I missed. You'll find by doing that, they've seen in studies, that individuals that have that kind of mechanism where they kind of put it away for a couple of days, come back to it and re-look at the material and try to recall it, they're able to make stronger connections in their brain. And that's this concept of presynaptic facilitation, plasticity, okay? Makes it easier for a neuron to fire. We'll look at some of the concepts when it comes to memory formation in the next lecture, or it's in the next chapter, I believe. I don't know if we'll get to it. I can inhibit a synapse. It's just an inhibitory postsynaptic potential. I don't really know how much I really need to get into that. There was glycine, it was GABA. Those are 2 main neurotransmitters involved with inhibition. And remember, it's just a hyperpolarization. Where's my thing? There it is. It's just a hyperpolarization. That's it. If we hyperpolarize, again, it's less likely to reach that threshold. And it doesn't fire. If we don't fire, okay, that could, if it doesn't fire, then we are inhibiting the release of excitatory from the next one. Calcium ion channels are inactivated. Okay? So I can inhibit these things. Pain is fascinating and interesting. You have receptors in your skin that are there to feel that mechanical event that's occurring there as I push down on my skin. Okay? But if I keep pushing, it becomes kind of painful. Right? So I can have an inhibition occurring to stop. I don't really, I don't feel the pain. I feel, oh my God, I feel the pain. I don't care about the touch. God dang, that hurts. So with pain will inactivate neurons that carry the sensation for mechanical touch. And it will release substance P that promotes the perception of pain. So pain gets traveled in that instead of mechanical touch. You've all witnessed this before. Have you ever tried to hang something up on the wall? Yep. Yeah. Sam, have you ever hung something on the wall? You, Gemma? Yep, Gemma has. Okay. And you're nailing that thing in, and then you miss that damn nail, and then what do you hit? Your finger, right? What do you do besides say naughty words? What do you do?
Pull back.
Pull back. You shake your hand, right? What the hell? Why are you doing that? Why are you doing that? Is it the wave sensation that gets rid of pain? No. I mean, this is mechanical movement, right? Or you rub it. You pound it with a hammer, and now the next thing you know, you're rubbing it really hard. Why? Because you're stimulating these mechanoreceptors. It's the same thing. The mechanoreceptor that sends that sensation of touch also sends pain up it too. So you can whack your finger there with that hammer, and then you sit there and you either do that or you rub it. And that rubbing helps to interfere with that pain receptor doing its part of sending substance P to the same dang neuron. And we can inhibit the action of substance P by just that mechanical rubbing of pain. Well, that's only for if you smash your hand. If it's an open wound and it's chemical burn, rubbing it is obviously only going to make it worse because we have a different receptor triggered there. Okay. But when it comes to this ability to inhibit, it can be a number of different ways. Okay. It could be whacking your finger and the pain receptor is overriding the mechanical deformation. That inhibits that mechanical sense of touch going up, and now all you feel is pain. Okay? Or by just rubbing that region that you whacked with a hammer with that mechanical deformation. I can rub that thing and I still have mechanical stimulation now, but now that mechanical stimulation is overriding that neuron that's releasing substance P. So a lot of fantastic concepts and ideas when it comes to, forming memories, causing pain to be perceived, inhibiting pain from being perceived. Okay? And all of it has to do with what in the world is happening at that synaptic level. We can facilitate it, releasing neurotransmitters to cause a greater increase in neurotransmitters being released, causing more calcium to go in, cause the synaptic vesicle to fuse to the plasma membrane, yada, 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 yada. I can do the same thing with inhibitory, okay? Where I can send inhibitory neurons in there. Less calcium entering, less neurotransmitters being released, less stimulation on the next nerve, inhibiting the nerve from firing. A much slower event. A lot of cool concepts. A lot of cool concepts when it comes to these things. This is going to be the basis for literally how everything works from here on out, guys. Skeletal muscle contraction, special senses, general senses, cardiac, the heart muscle itself, the nodal tissue itself firing, smooth muscle contraction, how we regulate the heart, how we regulate blood pressure, how we regulate the pH within the body, pH within the stomach. All of it is based on this nonsense right here. Okay. So we are never going to not talk about some of these things: nicotinic, muscarinic, ionotrophic, metabotrophic receptors. Metabotrophic receptors are G protein receptors. Okay. We're never going to not talk about these literally from here on out. So putting that time in now is going to make the other stuff a heck of a lot easier for you. I promise. Okay, I promise.